I do on my side, the radiopharmaceutical and radiochemistry production of PET. They're known as tracers that we make daily for our clinic and for research studies. So what PET stands for is Positron Emission Tomography. Actually, I enjoy giving talks like this to introduce people to the field. I remember we have dry ice and liquid nitrogen come in weekly. The one guy that came in and delivered us some liquid nitrogen says, "This is the PET center, but I don't see any dogs or cats around." So I like to, help people understand what PET is. It doesn't have anything really to do with animals, although we do animal imaging. So what PET stands for is a positron, which is the energy particle we're going to be looking at. Emission is the energy giving off, which in this case will be 511 keV or gamma radiation. And tomography are the images that we can collect, and we can slice through images to get a 3D picture of the person or subject being scanned. So I have the cartoon here from close to home. Great news, exploratory surgery turned up negative. With PET, it's considered non-invasive. So we don't need to perform surgery on an individual. The individual does get injected with a radiotracer, but it's a small amount and it has no pharmacological effect, that it's considered a non-invasive. So what PET is, what PET can be used for is for diagnosis of cancer, for heart diseases, and also brain disorders. So we're involved with oncology and psychiatry as well. So some uses for PET scans, as I mentioned before. For clinical uses, we're involved with the oncology group for cancer imaging. We also, on a daily basis, make what's known as nitrogen-13 ammonium chloride, which, if you know anyone that's gone for a nuclear stress test, this is the exact test that's given to the patient, where they're given basically radioactive ammonia-13. They're arrested, and then they stress them by either having them exercise or move around or breathe heavy for a couple minutes, and they're injected with the tracer again to see how well your heart valves work. If you're going in for surgery, to make sure that both chambers of your heart valve are working properly before you go through surgery. It's also used in the clinic, PET scans, to cross this magic thing known as the blood-brain barrier, for images such as Alzheimer's disease or D2, D3 receptors. We use the chemistry and the tools of PET in research to follow the drug pathway in real time. We're able to see the metabolic pathway of certain drugs. And because this is considered tracers and it has no pharmacological effect, you're able to see how this operates on the body without disrupting its normal systems. PET has actually taken off quite a bit in recent years. Not too far down the road from where I work at Columbia University Medical Center, New York City, down in Princeton, Bristol Myers Squibb broke ground about 3 or 4 years ago just to put in a PET center to aid them in their submission of phase 0 to 3 tracers. You can get a lot of information from radio labeling your tracer to bring it through a phase 0 through phase 3 clinical trials. So even the pharmaceutical companies are getting involved in PET pretty heavily. So on this image, this is, I don't know if you can see the whole screen. I can on my computer, but I'm assuming you can on yours. I got this image from Wikipedia, but it's a nice image in which it's a FDG scan. So this is what a PET scan will show you. What happens is a patient, in this case, is injected with F18 FDG, which stands for fluorodeoxyglucose. We'll learn a little bit more about FDG later in the talk. That's considered the workhorse of PET chemistry. What this does is in this case, I believe this was the scan for colon cancer. The FDG is a source of food for the cancer cells. It's a sugar cell with fluorine at the 6th position. So the cancer cells are hungry or more aggressive than your normal cells, they'll go in and eat this derivatized sugar molecule, and it gets stuck in the Krebs cycle, and it keeps eating and eating more and more of the sugar, and then it can light up what we call lighting up tumors inside the body. So this gives the radiologist a very strong tool into imaging cancer cells, into imaging what stage cancer you have, if you've been going through cancer treatment, has your cancer remissed, if it's spread or metastasized, and you're able to get these 3D tomography images in which we can take slices across the body to see in 3D what's actually taking place inside the body. The other thing that's really neat about PET is this is done in real time. So we can see, for instance, something enter and exit the brain in real time with the scans that are taken. And I'll discuss how these scans actually take place on the next slide. So how does a PET scan work? So this is what a PET scan looks like. It looks like an open MRI scanner. In theory, this could be a PET MRI or a PET CT scan. The PET scan just looks for the 511 keV radionuclide that had this mini nuclear explosion. It will not do anything to give you a background readout of your body. So what typically happens is a patient is put through a PET scanner, and it's in line with a CAT scanner or an MRI scanner to get the background of the body. So this way you know where the heart is, where the organs are. So how this works, the image I have here is we have a carbon 11 radionuclide. Remember from our periodic table, carbon 12 is the compound that we're used to seeing on the periodic table. It's a stable element of carbon. Carbon 11 doesn't like to be radioactive. It wants to give off its, in this case, a positron. This positron gets ejected from the nucleus to bring this down to a stable element. And it finds, sounds like it's a movie, it finds a negatron, sounds like a Transformer movie, but a positron actually finds a negative charge anion, a negatron, and it blows up in equal and opposite directions for energy of 511 keV. That's the magic number in PET chemistry. When this explodes and gives off the radiation in equal and opposite directions in this annihilation, 511 keV, all around this circular tube are these sodium iodide detectors. These are sensitive to pick up this gamma radiation or 511 keV explosion, you can think of it as. And what it does is it'll take that and it generates it as a registered number or a registered image. And the more and more that it sees at a certain time point in a certain area of the body, you get what I showed before, a glowing image of a cancer cell or whatever your nuclide was meant to attach to. So this is kind of a watered down version of how a PET scan actually works. Is everyone okay so far? Good? Again, if you guys have any questions, just please unmute yourself and ask me. I have no problem trying to field questions at any time. So originally, before I was saying that FDG is considered the workhorse of PET chemistry. So this is how a typical FDG scan works. If you know anyone that has gone for a PET scan, most likely due to cancer or something of that nature, 90% of the time they go for a FDG scan. And what FDG is a derivative of sugar, glucose, with a fluorine at this position here. It's actually not this orientation. It's a racemic mixture. They're not too concerned if it's alpha beta. However, we form this sugar, and this is what someone as myself or in my group, we would make on a daily basis for the clinic. So a patient may fast for 4 to 6 hours, and then they're injected with the radiotracer. A small amount, 2 to 10 millicuries, is administered. They're allowed to accrue this in their body, and they're put through the scanner. And anytime, for instance, in this one here, anytime something is picked up by our sodium iodide detectors in equal and opposite annihilation, it picks it up as an image. So this is how a PET scan works with the drug and the scanner. we not only use, we're not only using PET scans to image people. We actually, in our lab, this is a picture of one of our Micropet scanners. We use the Micropet scanners to scan murines or rodents, typically mice or rats. So you can see in this one here, this was, I forget, this might have been an FDG scan. You can see that the animal gets injected, and we can trace how well a compound works or a radiotracer works in a system. And this is very important because we use mice in research to help us as human beings progress in science. So mice are very, very useful. I like to give a slide and a kind of little shout out to the mice. They're very useful in the field of research. I don't deal with them personally myself, but I make the compounds for our biologists to work with. But we can do a lot with mice, and we can learn a lot from the rodents. So believe it or not, mice have very similar genome to humans. It's very cost effective and cheap to house mice. We can take a mouse from 0 to approximately 1 year old and get an entire lifespan of a human person during that time. So you can get a lot of data very quickly. We're able to grow mutations in mice. There's a lot of work done in mice, not so much rats, but mice to shut off certain cell lines and turn them on to get mutations. We can implant, unfortunately, implant diseases on them, manipulate mouse genome, but this helps us overall with the data we need before we can do any sort of study, including a PET study. So when we have a compound that we're bringing that's a novel compound, we would first look at cells and see what type of cell this particular compound may have an affinity to, say cancer, a pancreatic cancer over breast cancer. And then we can look at xenographing or putting this cancer cell line into an animal as a mouse and image it and see if it gets through the gauntlet of the body to get to a tumor to light up the tumor as a PET tracer. We also get a lot of information from the mouse, including the dosimetry. So what we really are concerned about in the PET field is not so much the drug itself, because it's in a small amount and we call it tracer amounts, that it has really no pharmacological effect on the human body. But we are concerned about how much activity is allowed to enter the body. For instance, if a lot gets taken up into the gut, you may only be able to inject 1 millicurie as opposed to 10 millicuries. And we get a lot of that information when we do rodent studies. We can extrapolate that data and see where most of our tracers would go inside a normal rat or mouse. So mice and rats are very important in our field and in all fields of sciences, I believe. So before I move on with what PET production is like, just to give a brief history, this is by no means a history course, but this is a brief history of radioactivity. So radioactivity has been around with us forever, but it was discovered by William Conrad Röntgen in 1895. I believe this month is Women's History Month. This is, if you ever want to read about women scientists, this is the family to read about. Mary Curie, also husband Perry Curie, and Henry Becquerel. And when we talk about radioactivity in the US and abroad, in the US, everything is mentioned in curies just because we need to be difficult. Everywhere else, they talk about becquerels. It's kind of like gallons and liters. But this is where the names come from. In 1903, they won the Nobel Prize in Physics for the discovery of radiation. So to win the Nobel Prize once in your lifetime is a huge achievement. Mary Curie won it again in 1911 and the Nobel Prize in Chemistry for her work in radioactivity. Not just to be done with his family, her daughter, Irene Eula Curie, won the Nobel Prize in Chemistry with her husband in 1935 for the synthesis of radioactive elements. So a very bright family and a very much involved in radiochemistry. Ernest Rutherford was the scientist who gave the names of different sort of particles. We have alpha, beta, gamma particles. For PET chemistry, we work with gamma radiation, which is the highest energy radiation. So a lot of the stuff that I work with are behind lead shields, lead bricks, really thick glass wall. So it takes a lot of time to build a lead wall. And then just to show, in 1939, the Nobel Prize in Physics was awarded to Ernest O. Lawrence for his development of the cyclotron particle accelerator, and he is a native of South Dakota, if you guys were not familiar with that. I know it's not the same. It's very close to you guys. I don't know if you rivaled with them, but that's kind of interesting that all around from our country and abroad, how PET and radiochemistry has evolved. So the way we measure radioactivity is an odd substance. You can't see it, you can't hear it, you can't smell it, you can't feel it. I guess if you're exposed to a lot of it, you'll be able to feel it, but by then it's too late. So the way we measure radioactivity is we use this thing known as a Geiger counter. Kind of looks like something from Ghostbusters. We have these lying around all over in the lab. Because we can't see, hear, feel, smell radioactivity, we need to check our radioactivity with a source, and we use what's known as this Geiger counter. These are the different ways to determine how much activity we're exposed to. And what we're concerned about, and I'll discuss this later on in the talk, is what's known as the topic of half-life. So half-life is the time it requires to basically reduce whatever you had to half of that. Each compound has its own unique half-life time. So carbon 11's half-life is 20 minutes. Fluorine 18's half-life is 110 minutes. Nitrogen 13's half-life is 9.8 minutes. Oxygen 15's half-life is 2 minutes. So there's a lot of different times, and there's pros and cons as to which isotope you'd want to use. As I said before, there's alpha, beta, and gamma particles. Alpha particles we can basically stop with, your hand. A sheet of paper would stop it. Beta particles you can use a thick piece of plastic, and you don't have to really be concerned with. And gamma particles, this will go through your body, so this is why we need to be concerned with it. But this is exactly how the PET scanner works. An individual is injected with a PET tracer, and the gamma radiation is able to travel through the skin barrier to these sodium iodide detectors found around the patient or the mouse that's being looked at. So we produce radioactivity inside our lab, but know that radioactivity is everywhere. It shouldn't be feared, but you should understand radioactivity. So just to give you guys some numbers so you understand about radiation, I always get asked, like, are my concerns for my life when I work? Yes, I am, but I check myself, and I'm actually less exposed to radiation than someone who does a lot of transatlantic flights. You're exposed to quite a bit of radiation when you go into the upper atmosphere. So the average radiation dose of a person is 360 millirems per year. You can just think of it as a number. So 300 is natural. So 60 millirems comes from additional sources. So some of the additional sources are, if you sleep next to someone, the potassium in that person is actually radioactive. Through a course of a year, you're exposed to 2 extra millirems. If you live in a masonry home as opposed to a wood home, you're exposed to 7 extra millirems per year. If you were to work at Grand Central Station in New York City, the employees don't know this, but they're exposed to an extra 120 millirems of radiation, which isn't that much, but it's still radiation per year because of the material that is used to build Grand Central Station. Another reason, too, if you are a smoker, to give up smoking. I don't know how much you have to smoke, how long you have to smoke, but you can be exposed to an extra 16,000 millirems of radiation a year if you smoke. Certain foods contain a lot of radiation, such as bananas, beer, Brazil nuts. If you were to ever look at purchasing a home, one of the things they have you look at, especially where I live in New Jersey, we do a radon check down in your basement to check for radioactivity. That can seep through your basement. They're actually able to estimate the age of the Earth based upon lead, uranium decay. So they estimate the age of the Earth to be 4.5 billion years, which is pretty interesting. And I'm not a geologist or anything of that nature, but it's pretty interesting that radioactivity decay is one of the sources, not the prime source, one of the sources that actually are used to increase the Earth's interior temperature. So you know how you have the crust, the mantle, the core? Radioactive decay is actually used to heat up the inside of our Earth, which I find pretty amazing. So now I'm going to get into what I focus on, and this is PET production. And the way I look at PET production is broken into three sections. We have the cyclotron, which is the initialization of our radioactivity. We have to make our radioactivity. Then we have the radiochemistry. That's to actually take our radionuclide and attach it to some sort of molecule to be injected into a person, a cell culture, or an animal. And then we have what's known as QC, or quality control, QA, quality assurance, where we check to make sure what we made passes certain stipulations before it is released to be injected into an individual. So this is, I took this image from number one to give them credit of the picture of the US. This is an image of all the cyclotrons from, I think it's 2016 this was done, of all the cyclotrons in the US that are used in PET production. There are 249 cyclotrons in the US. In New York City alone, we have 7 cyclotrons. Where I work, we own 2 of the 7 cyclotrons. So we have 2 cyclotrons in our facility. Actually, the closest cyclotron, oh, you guys have it in North Dakota, you guys have a cyclotron as well. There are over 1,000 cyclotrons worldwide since this number was taken in 2014. And I just put this up here to give kind of a perspective. It's not really as neat as it used to be when it was less than the number, which changed not too long ago. But Denmark, which is 16,755 square miles, 5.8 million people, they have 10 cyclotrons. It used to be 7 or 8. New York City, which is only 303 square miles with 8.6 million people, there are more people, but a lot less surface. We have 7 cyclotrons, which is kind of amazing. This is where we start to make our radionuclides. So radionuclides that can be prepared from a cyclotron. I discussed fluorine 18. Fluorine 18 is the one that's kind of kept PET industry marketable. So you can make an F18 radionuclide over several steps of radiosynthesis and be able to ship this to different areas. So you can have a place that just makes F18 FDG, and you can ship this as kind of like Amazon order wherever you need to send it within a certain time frame. It also gives the sharpest images because of its signal to noise background, and its gamma radiation is like I forget the exact number, 97% positron emitting. So it gives one of the best and sharpest images. Carbon 11, which is at our facility, we make quite a bit of, probably more so than any other radionuclide for research purposes. We make N13 ammonia about 8 or 9 times daily for the clinic, but it's not so bad of a synthesis, where carbon 11, each synthesis takes from start to finish approximately 4 hours, and it's just for one individual. The half-life of a carbon 11 radionuclide is roughly 20 minutes. This is nice because most of the compounds that we work with are organic, and what organic essentially means is carbon-based. So you're comparing apples to apples when you look at a compound that's carbon-based and you switched out a carbon-12 for a carbon-11. Whereas if you took a sugar molecule and you swapped out the OH functional group for a fluorine functional group, it's a different species now. The other compound that our facility works with is N13 nitrogen with a half-life of 9.7 minutes, and they do a lot of cardiac blood flow studies. Other compounds I've been involved with are zirconium-89, gallium-68, which is taking off quite a bit in the field of PET of recent. This is nice. It's actually the easiest one to remember. It's gallium-68 with a 68-minute half-life. It just works out to be that way. It's nice because it's created from a countertop generator, and it's enough to give a patient a single injection, and you can follow along, and you can start to label larger complexes of peptides and proteins, which are not typically able to do with a F18 or C11. And I'll get into that a little bit more as the talk goes on. Okay. So where do we get our isotopes from? From our cyclotrons. So this is actually our cyclotron. We have Siemens Eclipse HP 111 cyclotron. This is kind of neat. This is this cyclotron here. You guys can see it here. That's this guy here. Everything around it here is the shielding. This lead shielding, this is a picture I took inside our lab. This lead shielding is on tank wheels, and we roll it out on a tank wheel and we roll it back in on tank wheels. If this were to fall off the tank wheel, this would be going through several walls. So this is heavy duty stuff. We need this protection for ourselves because this is what we're going to be bombarding inside here, the cyclotron. So with the cyclotron, to kind of, I know my cyclotron engineer would get a little annoyed if I watered it down this way, but the way a cyclotron is made is just 4 parts to it. You have your vacuum system, your ion source, where you're going to actually get your radiation from and your energy from, your acceleration force, which is where you're going to, your RF, which is the energy that comes in, the power source, and you need beam control. We're going to have the beam control to make this a cyclic force and to control our beam to hit a certain window and a certain type of compound that we have in there, whether it be the target is water or some sort of nitrogen oxygen gas. Then we can make different radionuclides from that. So our cyclotron pulls, I wrote it down, our cyclotron pulls a vacuum of approximately 1 × 10⁻⁸ torr. To give you how strong of a vacuum that is, space is what I found online. Space is a 9.92 ⁻14 torr. So it's quite a bit off, but it's still, you know, it's to the -8 to -14. One of the Siemens cyclotrons at a nearby facility, this was years back, had problem producing F18, so we had to produce it for them for a while. They were down. They did a maintenance on their cyclotron. What it turned out was one of the cyclotron engineers at maintenance, the cyclotron, there's an O-ring in here. A piece of his hair fell and laid on top of the O-ring. And when they sealed the cyclotron, they weren't able to get vacuum. They couldn't figure out why. That's how strong and how delicate it is to pull this vacuum. Okay. So with our vacuum, we also get an ion forming source. This is what I talked before, our ion source. What we use is hydrogen gas. And what we do for our cyclotron, we take a hydrogen gas and it gets ripped off, or we put a proton on by ripping off on a beam of foil to give it an H-. The foil then strips, the foil strips the H-, giving us an H+, and what we form is a proton. And this is what bombards our target, a proton source. So this is how we make our radionuclidic nuclides. Again, I discussed that we need to have beam control. So this is where the cyclotron comes in. This beam control is controlled by magnets. These are known, these little Swiss cheese things here, there's these cheese blocks are known as D's and valleys, and they'll control your energy force, your beam, in a cyclic fashion, giving it more and more energy and more power. And when it hits its target, it gives it quite a bit of energy to bombard and basically knock off your nucleus of your compound you're working with. These are our targets we're looking at, or these are our targets we use. So if you go back to our cyclotron has this copper piece sticking out of the cyclotron. This is where our targets are located, and you can think of it as a gun revolver. Depending on what target we put in there, we can produce F18, carbon 11, or N13. This is what it looks like here. And we can fill our targets up with whatever compound we need to use. So this is how we generate our radionuclides. And now we need to attach these radionuclides onto a parent molecule or a parent compound, which we know goes to a certain area of the body or a certain type of tumor that you could think of for cancer cells. And this is where radiochemistry comes in. And this is what I'm heavily involved in, the radiochemistry. So everything we do is work behind lead shields. These are some of the lead shields. This isn't particularly our group, but this is what some of our lead shields look like. We run things automated and some manual. We need to use these mechanical arms to work manual labeling. And timing is everything. We need to have everything down to a certain time frame with our clinic, our production team, our QC team, and our cyclotron engineer for when we need to bombard stuff. A lot of these compounds are quick to decay, so we can only be done in 1 or 2 steps, and we rely on HPLC for purification and identity. We're not able to bring this to an outside source to run like an NMR or something on our compound. Plus, we make such a small amount. We take an HPLC reference profile and compare it to our radioactive compound. So this is what a lab may look like, where you see they can do some manual manipulation with these arms. This is a guy standing, this is on the inside of a hot cell. This is him standing on the outside. You can use these arms to manipulate your syringes, to move lines around, or you would have an automated synthesis box. So carbonyl, I'm not going to get too into the chemistry involved, but I have it up on the slide here to go through it. And we could just see by looking at where the red carbon goes. So carbon-11 chemistry, we've done Grignard reactions where we can attach this carbon-11 atom inside a molecule. And this compound is known as C11PHNO. We do quite a bit of PHNO studies in our lab. We also do electrophilic methylation, which is traditionally the type of chemistry carbon-11 is used for with C11 methyl iodide or C11 methyl triflate, where you have a very strong nucleophile and you have a good electrophile. So you go back to your undergrad organic chemistry, nucleophile finds electrophile, negative finds partially positive. It attacks it, knocks it out, and you're left with this radioactive C11. This is known as PIB. It's a Pittsburgh B compound. For those that work in the field of C11, we call this C11 PIB for pain in the butt because it's kind of a pain to make, but it's actually Pittsburgh B compound. There's also nucleophilic cyanide reactions where we can make radioactive cyanide gas, which sounds very, very harmful, and it is. However, you can start doing some building blocks of small chain amino acids. This is a project I was working on, a glutamate project, on which we make this glutamate species by taking a radioactive cyanide group or HCN group, attaching it to the molecule, and then in this case it'd be oxidizing, reducing, however you want to look at it, oxidizing the molecule to form your amino acid derivative. So the thing that I just want to point out with carbon-11, so this is what one of our boxes look like, a close-up of the box, but we won't get too involved with it. And this is how we transfer carbon CO2 into our C11 methyl iodide or C11 methyl triflate. But what I'd like to discuss with carbon-11, it has the identical properties of what we see on the periodic table, carbon-12, physical, biological, chemical, and pharmacological. So carbon-11 is used in research to ensure that certain drugs are metabolizing without any harmful byproducts. So a lot of the drug companies, in particular Bristol Myers Squibb, that I know down in Prince, New Jersey, broke ground a couple of years back for a cyclotron facility. And this is what they are using it for: radiolabel small molecules without altering its physical, biological, or chemical state, and they're able to test how this works in real time inside the body of either cells, animals, and potentially human beings for their drugs to bring them through phase 0 through phase 3 clinical trials. Just going to show you a couple examples of carbon-11 compounds. This is a PHNO study and a raclopride study. These are the two different compounds. The cyclotron makes the same carbon atom that comes out of the cyclotron, and then the radiochemistry manipulates it to make it into the molecule you see above or the molecule below. And you can compare the two. And in this study, they compared PHNO versus raclopride and showed that PHNO is a better D3 agonist. It's used in antipsychotic treatment for blocking D3 receptors. The other radionuclide that we work with in our lab, not so much as carbon-11, is fluorine-18. The half-life of fluorine-18 is 109 minutes. You can do electrophilic or nucleophilic reactions with F18. I've personally never done electrophilic reactions with F18. We've done only nucleophilic reactions. The nice thing with F18 is a cyclotron can produce large amounts of raw F18, and this allows you to synthesize molecules and send them out to other locations, so you have a satellite center and you can send it out to different hospitals. Fluorine-18 has some unique characteristics, but for our radiochemistry, the half-life of 109 or 110 minutes makes it very cost-effective and very useful in studies in a clinic, because you can take a scan for up to an hour and a half sometimes if a patient is able to lay that long with an F18 scan. One of the things that came out of the synthesis of FDG or F18 is the use of what's known as cryptans or Cryptofix. Just to show you another field that wasn't really involved with radiochemistry, is now a mainstay in radiochemistry. This cryptan or this Cryptofix ligand, if you decipher what the word crypto or crypt means in Latin, it means a place to die, like a crypt. What it does is this cryptan forms a 3D chelator around your potassium ion, and it sucks it away from your F18 molecule, making it more nucleophilic, or what they call naked or free, to react with. So this type of chemistry was awarded the Nobel Prize in chemistry in 1987 to Cram, Ley, and Peterson for their work on Cryptofix or 3D crown ethers and their analogs. So some of the radiopharmaceuticals used with F18 are what I said before, is fluorodeoxyglucose. Like I said before, FDG is considered the workhorse of the PET industry. There's also, you can do bone scans with sodium fluoride. And where Dennis had discussed in the opening that I worked at, originally I was working in my postdoc at UPenn under the guidance of Professor Hank Kung. His group was the group to make amyvid or fluorobetaper, and this is now an ANDA regulated compound for Alzheimer's imaging scans. So it's kind of neat that I joined the group. They already had another company across the street in Philly working on this heavily, but I was part of the group while we were in the works of getting this submitted for an ANDA, which is just an acronym for a new drug application. So the insurance companies can reimburse, and the patient can be given this type of treatment or imaging out of the cost for the doctor with what am I trying to say? With insurance purposes. So it's an approved drug, or in this case, a radiotracer. Another compound that our group makes and is used in imaging is F18F-DOPA. This is interesting because it crosses the blood-brain barrier. So this is the images of the brain. Again, remember, PET is a positron emission tomography. So even though the patient was scanned lying down, we can do cross sections of the brain tomography to get a better image of what's going on in the brain. And you don't have to cut an individual up. You can do this through your imaging slides. So this makes it a very unique and powerful tool for imaging. F-DOPA is used to image neuroendocrine tumors through the amino acid transporter chains. So another compound that we make quite often is N13NH4Cl, which is we call it N13 ammonia. It's used in our clinic typically for a nuclear stress test. Here you can see a healthy individual when they're rested, given a shot of N13 ammonium chloride, and then when they're stressed, their valves are working properly. And this individual, when they're resting and stressed, it's not the same. So this person may be considered a risk factor when they're going for a surgery. Originally, technetium-99m was used. I've not seen, maybe it was not around when they were doing TI-201, but technetium-99m was used, and a lot of the cardiologists now are switching to N13 ammonium chloride for the ease of making, and also there's not as much technetium-99m available. The half-life of N13 ammonium chloride is less than 10 minutes, so this thing just goes away fast. As I said before, gallium-68 is another compound that a radio tracer that is used in this case for a prostate-specific imaging, PSMA. You can look at tagging large chain or bigger chained compounds with a gallium, with a metal. F18, if you were to look at the chemistry, it's a very harsh nucleophilic basic reaction. It would rip apart your peptide sequence. C11 is very hard to add at a specific site without doing multiple protection, deprotection steps. So the next best thing is to make these dotatate or dotatok type of chelators, and it fits in nicely with a gallium or some sort of metal, in this case gallium-68, and you get a nice PET imaging agent, as you see here. They compared gallium-68 to F18 choline, C11 choline, and FDG. So it's just another tracer that's used in the PET production. So lastly, what I want to discuss is the quality control, quality assurance. So our lab is regulated by the FDA. We get audited by quite a bit of entities, the FDA being the supreme power of all of them. Before anything is released, it needs to go through a quality control measurement where we check for radionuclidic purity to make sure we have 511 keV. So the cyclotron sent us what it was supposed to send us. We check for radiochemical purity to make sure that we have no other radiochemical impurities involved in there. It needs to be a certain criteria. We also check for the chemical purity. So whenever we make a carbon-11 compound, there's some carbon-12 that comes through with it, which is known as what we call the cold mass, or that's the actual mass of the drug. That carbon-12 amount, which we normally measure in micrograms per mil, that, if anything, gives an adverse side effect to a patient would be the cold compound, because that would attach to the receptors and there'd be a measurable amount of it. You cannot measure how much radioactivity you make. I had to wrap my head around this when I was beginning of this process of radiochemistry. You can't put it on a balance and say I have this much radiochemistry. You measure it in a gamma counter, so it tells you how much activity you have, but it's not measurable how much you actually make. We also look at the mass of the impurities that's made through HPLC. We do a pyrogen test, which checks for any sort of excipients, or not excipients, any sort of foreign matter that if someone would be injected, they'd spike a fever. So we do a pyrogen test. That test is pretty neat. They use a horseshoe crab blood to run the tests, and this is a Charles River company out in South Carolina. But they literally will have a farm of horseshoe crabs, and they pump the blood out of them. It's pretty neat that these horseshoe crabs are able to do that. We have to perform sterility in our compound, but because we fall under certain guidelines, our sterility isn't performed for 30 hours later. Even though we've administered a drug, the patient has excreted the drug and it decayed within a couple hours, we're allowed to do our sterility 30 hours later for safety of our workers. Now, it's kind of an odd rule, but we still have to follow. Anytime we inject the patient, We follow this drug for another 14 days to make sure everything passes with sterility. We look for residual solvents with our GC, and we check for the pH. And then we have a quality assurance chemist or a pharmacist check over all the data that the QC person did to check the production person. So we have a lot of checks and balances in the lab. How am I doing on time? Okay, so I'm almost done. So the people that are involved with PET scans, you don't need to be involved in the PET chemistry area like myself. You could be involved as the primary care physician to recommend the scan. We work alongside psychologists, neurologists, and oncologists. My boss, I guess not really advisor, my boss is a radiologist. We work with surgeons. One of the members of our lab is a nuclear pharmacist. I also work with the nuclear technicians over at the clinic. Then we work with the nurses as well. I work with the cyclotron engineer. There are also biologists and veterinarians that are involved in our work, health physicians, radiation safety officers, and then for research-wise, mathematicians and molecular modelers get involved in the PET imaging. Other areas involved in PET, I'm considered a radiochemist, and who I work with on a regular basis, as I discussed before. Internally, there are other groups that we work with. IACOOK for veterinary purposes. Equipment, we actually work quite a bit with facilities. We have a chiller pump we have two chiller pumps, one that chills our building and one that chills our cyclotron. So our chiller pump is the same size as our building chiller pump, which is crazy. But we work with facilities quite a bit, Siemens and GE, LabLogic and PerkinElmer. And then for audits, we get audited by everyone and their brother, it seems. The FDA, the DEC, the DOT, the EPA, New York State and City, the FDNY. A lot of these guys and girls are very nice to meet. They're trying to understand us. We try to understand what they're doing. So it's actually a very good meeting with them most of the time. It's not, beware, they're here. But it's kind of neat to see how, because they're enforcing laws on us and we're explaining to them what we're doing, and we have an open dialogue with them. So it's pretty neat to see. But you can be a Department of Environmental Control, and you could be working with a PET center. You wouldn't even know that. But they're involved a lot with the PET industry. So there's just a picture of myself working in one of the hot cells. These are the arms here. This is an ammonia production. So I love working in my job. We develop new drugs. I work on new compounds on a monthly basis. I'm given free range to design my own equipment and experiments. I work with great people with great diverse backgrounds, biologists, pharmacists, engineers. It's nice because I feel like I'm making an impact on society for people who've gone through either cancer treatment or are, going for a cancer scan, and it has a positive impact, I feel. So where I see the future of PET chemistry going is there's plenty of new ligands out there to design for specific disorders. We work with a lot of automated processes, kind of how the Keurig has turned into people don't know how to make coffee anymore. I think that's where we're going to go with PET chemistry. It's going to be a plug and play where they're not going to really, it's a good and a bad thing. Won't really know how to make a PET compound, but everyone will be able to make it through automated synthesizers. I like doing things a lot manually, but we do have a lot of automated synthesizers. In the field of PET itself in chemistry, there's a lot of methodology I feel that needs to be worked out. We have the luxury, and it's nice that we're never really hurting for money because we have a clinic that we supply, but we never have time to do real research. And I don't know if the field of PET chemistry has that. That's something that may come around soon, but there's really no methodology worked out in PET. It's kind of just beat it with a hammer to get it to work. And as long as it works enough for an image, it's workable. There's a lot going on in the field of radiotheranostics, where you have an imager and a therapeutic to do the chemistry. So you can image and do the therapeutic with a radioactive compound. So that's something that I think is going to have a future in PET chemistry. Right now, the stuff that I work with is just for imaging. It has no radiotheranostics. Other isotopes that are, these have always been on the market, but gallium-68 is getting a big push again. I think it's because it has to do with the radio labeling small amino acids, and it's done on a tabletop generator. Targeted therapy, and then more ligands will be approved by the FDA through what I discussed before, NDAs. So just to, end here, this is where I work. So this is the George Washington Bridge. If you're ever in New York City, you give me a shout. I can give you a tour. This is where I work, 12 floors below ground. They don't let us out too often, but no, I'm kidding. So this is my group. It's myself here. This is our cyclotron engineer, Simon, our radiopharmacist, Mark, Dr. Oya, and Dr. Bott are our other radiochemists. Sydney's our technician, and this is the head of our department, Dr. Mintz, the radiologist. And then we have a couple biologists. I work with these guys as well. A couple biologists and radiologists in there, but this is our core team here. So just want to give thanks and acknowledgments to the people I've mentioned before. And I especially like to give thanks to the Visiting Science Series, especially the people that helped me set this up, and Dennis as well for inviting me out to give this talk, and to thank you guys. If you have any questions, I'd be happy to answer them. Thank you.
Thank you, Dr. Carberry. There were some great questions. There's actually one in the chat. How much radiation do you think you would be exposed to in a year?
Oh, so we get tested, and actually we just had an audit from one of the Department of Energy come to do an audit. I'm well within below what's considered below ALARA 1 for a year. So there's different ALARA levels. So I'm not any much more than the average citizen. We wear what's rings and badges. So what we do is we wear rings and badges, and it keeps track of how much activity we're exposed to, and we are audited internally by our radiation safety, and every month we meet with them and go over our previous month's rings and badges. If we pass ALARA 1, we'll get that's okay to go past ALARA 1. We'll get a notification in your ALARA 1. If we make it to ALARA 2, they would interview us and make sure what we're doing. We'd have to change a process around. Maybe we're exposed. We have to go into a hot cell too much with our rings and badges. If we're exposed to ALARA 3, which would be higher than the normal citizen, we would be sit out. So we wouldn't be able to be exposed to any more radiation. So we'd have to do like desk work, paperwork. So we're monitored quite a bit, and I'm very comfortable where I work, and I'm very safe with what I do. So I'm not too much more radiation, maybe 400 millirems per year as opposed to 360 millirems per year to the average citizen. That's a good question.
Thank you.
Are there other questions?
I don't know if you mentioned this, but who invented the cyclotron, or how did that come about?
Yep. If I go back to, well, I just ended the slide, but yes. So the cyclotron was invented in 1939 by Ernest Lawrence, who was, I guess he saw a diagram. I was reading about this about a week ago. He saw a diagram of a potential cyclotron particle beam accelerator just on paper. Someone came up with an idea, and he actually took it to heart. He's a South Dakota native. He took it to heart, and actually the Lawrence Berkeley Center in California is named after Ernest O. Lawrence. So the National Laboratory in Berkeley is named after him. But he developed the first particle beam accelerator in 1939, and it hasn't changed too much. It's been a lot more efficient, but it hasn't changed too much overall design. So it's a pretty noteworthy development.
I have a
And,
question, Kat. Oh,
Yes, go ahead. Go ahead,
Go ahead.
Dennis.
so what are typical properties of compounds that you can use as, radio tracers? Because it seems like there are a variety of compounds that you guys have been using. It could be, an amino acid, it could be a carbohydrate, but is there any specific, type of molecules that you can actually use, something like that? Or is it, are you considering that these molecules have to be water soluble as well?
Oh, good question. So there are the answer that I would give is any compound you could think of to make, you can make as long as you can add your radio tracer to it. But what we'll typically do is, for instance, one of the compounds that we just got approved, and we had our first human injection about a month ago. The compound is called MPC. It's a microtubule cancer agent, so it destroys cancer inside the cell. And it's been used as a cancer therapy agent, but no one's ever radio labeled it. So we radio labeled, because it's an O-methoxy group, we radio labeled the O-methoxy group at C11, and now we use it to image to show it past the blood-brain barrier and to show how it would actually attack a cancer cell. So you can look at a compound that's out in the literature or on the market, and you can say, hey, I can radio label this because it has a functional methyl group for carbon 11. Or you can swap out, like, for instance, an alcohol group and put a fluorine group there. You do change some of its properties, but that's a way that you can go about thinking to create a library of compounds for radio labeling. And then the typical way to go about and test a drug, or we consider them radio tracers. They're not really drugs, they're tracers. You would take whatever scaffold you want to make your compound. Say it's amino acid with a fluorine on there, or it's a small aromatic ring with a carbon on there, and you would radio label it and subject it to different, if you're looking at cancer cell lines, subject it to different cancer cell lines and do a binding study where essentially you let it incubate on a bunch of cell lines, you wash it off, and then you throw it through the gamma reader again, and you see how much of this radioactive tracer sticks to it. And it's easy to tell because you can put it through a gamma reader. So if you start with 1 microcurie of sample, or say you start with 1 microcurie of sample, you wash it off and you put that same tray through the reader, and you're left with 500, or say, sorry, it's like half of that. Then you know that it has half binding affinity, which is pretty good. The next step would be, once you find, say it was very specific for breast cancer cell lines, you would then take a rodent, a mouse, for instance, and you can grow a tumor of breast cancer cells on that rodent. And then you can inject the rodent and see if it can bind to the same cancer cell lines which you forced onto on a petri dish. See if you get through what I consider the gauntlet of the body to make it to the tumor. Say they put the tumor in the shoulder, see if it gets bound to the tumor on the shoulder. This shows you that the compound can make it through a body, because what the body wants to do is take this, whatever foreign object is, and break it down into small amino acid chains so we can use it for energy. So if it can make it there and bind to the cancer cell line, you have something that's worthwhile. Then you would, after you did a bunch of studies on rodents, you could do dosimetry studies and uptake studies on rodents. You would move to larger animals. At Columbia, we have not me, but the groups associated with us have rhesus monkeys, baboons. So you'd use a primate to look at it. You can look at pigs, because pigs have quite a bit of the same gut area and cells as humans. So you could look at a higher chained animal, and then you would apply through the FDA for like a phase 0 injection, where they give you essentially what my old advisor used to call 10 freebies. So you get 10 injections to check out the dosimetry of your molecule. You would take 5 normal men, 5 normal women, a certain age or demographic, and you would look to make sure the drug or the radio tracer doesn't have an adverse side effect on these volunteers. And then from there, you can submit it to the FDA as a IND, investigative new drug. where you can look at and write a protocol or a grant out to study whatever you're looking at, metastasized breast cancer in, I don't know, women. So then you would do a study on metastasized breast cancer in women using your novel drug and compare it to something else. So it would start in a petri dish, move to animals, larger animals, then to humans. But it all starts from essentially whatever you feel would work. You could go to the literature and look and see what's been made in the literature, or you could develop something that no one's developed before.
Sounds good. I think Aaron actually asked a question. So his question is, do you think regular metabolic activity, or is regular metabolic activity radioactive? If so, do you believe that PET scanners could become sensitive enough to detect that on its own without the use of radio tracers?
So I'll answer. Let me see if this answers your question. So what we do with our tracers is we would inject, if it's a new tracer, you inject the tracer, and then at certain time points, 0 minutes, 10 minutes, 30 minutes, you would put an A line in the patient and you would draw blood. And then the tech would spin the blood down, do accounts of the protein, and then extrapolate the organic compounds from the blood, whatever means protocol we have to do that, and shoot it on an HPLC, a high performance liquid chromatography, where you get a trace. And then what you would want to see is with your radioactive probe, you would see your parent peak. And then if you see any other peaks that are associated with it, you would consider those to be metabolites of that parent peak. Depending on how involved you get with the study, you'd want to start isolating and characterizing those daughter peaks of the parent peak. Those are considered your metabolites. So for instance, like with, I think it's Tylenol. Tylenol itself is the acetate, but the actual carboxylic acid is the one that provides the pain alleviation. But if you were to take the carboxylic acid form of it, it wouldn't reach it to your receptors. So it needs to actually make the metabolite to get to that part of your body. I believe it's Tylenol. I could be wrong. With our PET tracers, we want to see if it breaks down into some other metabolite, because you might be getting a false reading. Or it might be that, hey, I got a lot of uptake after 2 minutes, but then it went away. What happened? Well, maybe through taking traces out of the blood sample, this has metabolized to form some other radio tracer, which is not active in that site of the body. Does that answer your question?
I'm not sure. Aaron, do you have okay. He said thank you.
Okay.
Do we have any more questions from the audience?
And if you guys have questions and don't want to ask now, you could always send me an email. I don't know if you saw on the last slide my email address, but I'd be more than happy to answer questions via email as well.
I can share with my students your email address.
Sure.
And they can send you a message if they have any more questions. So, but anyway, well, if there's no more questions from the audience, then we thank you for giving us your time to give us this presentation, and I hope we my students in general have gained something from it, especially they do study organic chemistry with me. So
okay, great. Thank you so much for giving me the opportunity to speak, and thank you so much for having the patience while I couldn't get my computer to work. I had to jump on my wife's.
Thank you, Dr. Carberry. We're getting a lot of thank yous in the chat, so we very much appreciate your being with us. All right. Thanks, everyone, for being with us. Good night.
All right. Thank you. Take care.